So, today we are going to start with the gene and protein synthesis, right? First, let's see what are genes. As in the beginning, in the previous lessons, we have discussed that gene is the fragment of DNA, fragment or a small base sequence of the DNA, fragment or base sequence of DNA. DNA stands for deoxyribonucleic acid, right? So gene is the fragment or the base sequence of DNA that is used to form or to express a particular gene or specific protein, particular protein, right? Now we will also see that how the DNA base sequence is used to make a particular protein and what determines that how the different proteins are formed from the different types of genes. For example, let's talk about the genes first, right? In genes, you know that genes are present on DNA and DNA has got a double helical structure, somewhat like this. And if we open up these two strands, you know that each strand is made by nucleotides. What are nucleotides? Nucleotides are the basic unit of DNA strand, right? And this one nucleotide contains a sugar, right? And a phosphate group. You may have studied before in the chemicals of life, biological molecules, where we always talk about the carbohydrates, proteins, fats, and the fourth biological molecule that we have talked about was DNA and the structure of the DNA. Right? So I'm going to revise a little bit of the structure of the DNA first, and then we will move on to the protein synthesis so that you understand it well. So a nucleotide contains a phosphate group, a sugar, right? This is a sugar, right? It's a pentose sugar because it has got a five carbons in it, right? And on the other end, it has got a nitrogenous base. And nitrogenous bases can make the nucleotides different from one another. They can be a nucleotide with an A. There can be, instead of A, if there will be a T here at the same position, right, in the same structure. So we can say it's a thymine. A is adenine, and there can be a guanine, right, a sugar, right, with it, a guanine base and a phosphate group, right? Another nucleotide. There are four different types of nucleotides that can make DNA, right? That can be present in the DNA molecule. And you know that they are complementary to one another as well. These two lines that I have made here to make the two strands of the DNA, these backbones are made by sugar and phosphate and the nitrogenous bases, that this is A and T and G and C. They are hanging in between these two strands and they are making up some bonds between them, and they also show a complementarity. What do we mean by the complementarity? That is, A can only be in front of T, and T can only be in front of an A, right? So this is the complementary base pairing, we can say. And G can always pair up with C, and C can always pair up with G, no matter whatever the sequence of the gene is, right? Obviously, I have made a small fragment of the DNA, but genes can be bigger, right? They can be made by hundred and thousands of the base sequences, right? And those base sequences could be different for different genes. So this is something that makes proteins different from one another because these genes have got genetic codes in them. And these genetic codes are always triple code, right? For example, these are the two strands of DNA, right? Let's suppose there is a sequence on one strand A, G, C, C, A. It could be different. I'm just writing randomly, right? A, T, T, C, C, G, right? So there can be different base sequences present on one fragment, and they make different type of protein. For example, on another fragment or on another strand of the DNA, there can be a different sequence of the gene, right? That will make a different type of protein. So these are the genes that the base sequences that make a protein different from one another. How? You know that DNA is DNA. DNA is present in the nucleus, and the proteins are made by amino acids, right? So these are the genes, right? Now the question arises how the genes are converted into proteins, right? And what determines the sequence of amino acids? The sequence of amino acid is completely dependent on the sequence of the bases present in a gene, right? For example, for this base sequence, there can be a different sequence of amino acids. For example, let's suppose I'm just diagrammatically presenting you different types of amino acids in these shapes, right? Let's have a look. Right? So there are four different types of amino acids that are present, and this gene is used for these four amino acids to make a sequence like this, let's suppose. Right? This is the sequence. There can be amino acids repeated in a sequence like this, right? Like this. So this is one sequence of the gene. There can be a different sequence, like suppose like this. This protein and this protein would be different. These are the two different proteins that I have made here, and they are different because they have got a different sequence of amino acids. And this is protein A, this is protein B. Why they are different? Because they have got a different sequence, right? This protein maybe is made by this gene sequence, and this protein maybe is made by some other gene sequence, right? Or the base sequence that is present on some other chromosome. So for every different proteins, we have got a different base sequence present in the DNA that makes the protein sequence different from one another. This is how the proteins are different, right? Now the question arises that how this happens, how the genes information that is present in the nucleus, are you getting me? That is present in the nucleus is converted into a protein amino acid sequence. Now let's have a look on the proper process of protein synthesis. Right? Now we will see the second part of the today's lesson, that is protein synthesis. Protein synthesis is basically protein means that making an amino acid sequence. It happens in two steps. The first step takes place inside the nucleus, in which the nucleus that carries the genes or the DNA base sequences are first converted into mRNA or messenger RNA. This first step that takes place inside the nucleus, this is sometimes also called as transcription, right? Transcription is the process in which DNA's information is converted into mRNA, right? mRNA means that this is the polynucleotide sequence or base sequence, or you can say a polymer of RNA nucleotides. There are two different types of nucleotides you have studied in the chemicals of life. When you studied the DNA structure, you also studied the mRNA structure. I will give you a little detail of these two structures in this lesson. The second step is translation, right? Translation is the process that takes place in the cytoplasm with the help of ribosomes, right? And those ribosomes are attached cytoplasm, right? And particularly on RER. RER stands for rough endoplasmic reticulum. Rough endoplasmic reticulum. This is an organelle, a very important organelle that is involved in protein synthesis because they have got ribosomes attached on them, right? And these ribosomes are actually involved in making a uh, peptide chain, right? That is a protein. So this transcription takes place inside the nucleus where the DNA is present, right? And the DNA is converted into mRNA. And then that mRNA is a polymer, is a chain-like structure that is made from DNA, then comes out in the cytoplasm from the nucleus through nuclear pores and then attaches itself on the rough endoplasmic reticulum where the ribosomes are present. And then these ribosomes actually make the polypeptide chain. So the basic process briefly is this, right? First step that takes place, that is transcription. The second step is translation that takes place outside the nucleus. Now let's have a look at how it is happening, right? With the help of a diagram. So this is the DNA molecule, right? The double-stranded molecule that has got the information for the development of a particular protein. First, this DNA molecule gets open, right? And this DNA molecule has got a base sequence, right? A, T, T, G, C, C, G, whatever the sequence is, C, right? It can be in repeated form, right? And what happens is from this base sequence, there will be a messenger RNA that will be formed in front of this DNA according to the complementary base pairing. In front of A, there can never be a T, there will always be a U. Because in RNA, because this is messenger RNA that is being synthesized, and in the RNA, there can never be a T present because it cannot make the bonding with an A, right? So they instead of a T, there will always be a U wherever there is an A on the DNA molecule, on the DNA strand. With T, there can be an A. With T, there will be an A, right? And with G, there will be a C on mRNA. With C, there will be a G, G, C, and G, right? And then this messenger RNA comes out, right? Comes out in the cytoplasm. And then in the cytoplasm, this messenger RNA is grabbed by a ribosome, right? And when these ribosomes will move on the mRNA, they will make a polypeptide chain, right? So there will be a polypeptide chain made by the different types of amino acids, right? And it keep running and it will keep making a polypeptide chain, right? So this is how the first step that takes place in the nucleus, right? That is transcription in which the mRNA is formed, and then it comes out in the cytoplasm and attaches itself with RER ribosomes, right? And these ribosomes basically help in making a polypeptide chain. We are not going in the details that how the tRNA comes, how the ribosome helps in making the peptide bonds, right? Because in IGCSE, we will stay confined to this that first you should know that mRNA is formed in the nucleus, and then mRNA comes to the ribosomes where the polypeptide chain is formed. So this is the poly peptide chain that is formed, right? Chain that is formed in the cytoplasm on rough endoplasmic reticulum. And then this polypeptide chain that is made by the different amino acids, this sequence of these amino acids that is made here is dependent on the base sequence that is present on mRNA. And its sequence is dependent on DNA base sequence, right? So this is how a protein that will be formed from different amino acids joined together in a particular fashion or in a particular sequence that makes a protein different from other proteins. I hope that's clear to you that how the messenger RNA, how the protein synthesis is taking place. Let's have a brief recap. So first, what is a gene? Gene is the fragment or the base sequence of DNA that is used to form a particular protein, right? And we have studied about the DNA structure first. That DNA is a polymer of nucleotides. Nucleotides are the basic units, you can say, for the formation of a DNA. And DNA is a double helical structure, and both the strands have got the nucleotides, right? And those nucleotides are attached to one another with the help of hydrogen bonds between them, right? Here they can have two hydrogen bonds between them, an A and T, and they also show you complementary base pairing between them, right? A G and C with G. And they always, you can say, are attracted towards one another by making the hydrogen bonds. Bonding is always a force of attraction, right? So these nucleotides are present in the DNA two strands and they join together by making hydrogen bonds, right? So next is that this is a gene and how the proteins are different from one another. That is according to the base sequence that they have got in genes, right? And we have got only 20 different types of amino acids discovered so far, right? And we can have thousands of different types of proteins in different individuals, right? So how come that there are thousands of different types of proteins? That is basically the sequence of the protein amino acid sequence, right? That makes a protein different from one another. I have just told you that how this A is different from B, right? Because of the sequence of the amino acids, right? And that is determined by the sequence of DNA nucleotides, right? Or the base sequence that is present in the DNA. Now the question arises that how the protein synthesis takes place. This takes place in two steps. The first step takes place in the nucleus, and the second step takes place in the cytoplasm on rough endoplasmic reticulum. In the first step, the mRNA is formed from DNA. I have just explained you how this happens in the nucleus, and translation is basically the process in which the polypeptide chain is formed with the help of ribosomes, right? And the more of the details are present in your A levels section, right? So we will have a look on the detailed, you can say, the process of protein synthesis in A levels when we'll study A levels protein synthesis, right? So I think that's it for today.